Hello guys, I am Sagar Sonone and today I am going to discuss about the central dogma of life. Okay, so what is central dogma? So central dogma is nothing but central dogma is a molecular biology that deals with the detailed residue by the residue transfer of sequential informations. Okay, means in the central dogma the sequential information can be transferred. Okay, it states that such an information cannot be transferred back from the protein to either protein or the nucleic acid. Okay, so in the central dogma, uh, DNA to RNA, that is a transcription, and RNA to protein. Okay, means RNA can made up of RNA can be made of protein, that is a translation, means the synthesis of the polypeptide specified of an mRNA, that is translations. Okay, so protein information can be flow back to the nucleic acid. Okay, and the fundamental framework that understanding the transfer of the sequence information between the by polymers. Okay, the central dogma of a biology is that the information stored in the DNA and transferred to the RNA. Okay, during the transcriptions of to protein during the translations. Okay, so DNA to RNA, the information can be carried as a genotyping, and RNA to protein as a phenotyping. Okay, and for requirements for the DNA to RNA, it means DNA to genetical material. Okay, so must carry information cracking the genetic code, must replicate the DNA replication, must allow for the information to change, that is a mutation, and must govern the expressions of the phenotype, that is a gene functions. Okay, so DNA replication, the process of replication to the entire genomes prior to the cell divisions. Biological significance, the extreme accuracy of DNA replications is necessary in order to preserve integrity of the genomes in the successive generations. In eukaryotic, replication only occurs during the S phase of the cell cycle, and the replication rate in the eukaryotic is slower, resulting in a high accuracy of the replication in the eukaryotes. Okay, then the basic rules of the replication, that is a semi-conservative, starts at the origin. Okay, synthesis L1 5' to 3' direction can be uni or the bidirectional, semi discontinuous, and last is the RNA priors required. Okay, so semi conserve means one strand of duplex pass on the unchanged to each of the daughter cells. Okay, then next, that is the starts its origin, mean intended protein identify specific base sequence on DNA called site of origin, and the synthesis is always in the 5' to 3' direction. Okay, then next, uni or the bidirectional means replication forks move in one or opposite direction. Okay, semi discontinuous replication means anti-parallel strains replicated simultaneously, leading strain synthesis continuously in 5 to 5' to 3', and lagging strain synthesis in the fragment in the 5' to 3'. Okay, and semi discontinuous replication, that is a new strain synthesis is always 5' to 3' direction. Okay, core protein at the replications. Then the next is that the mechanism of the DNA replication. Okay, so there are the three, that is the intentions, elongations, and terminations. So first, that is in intentions, proteins bind to the DNA and open up to double helix, prepare DNA for complementary base pairing. Elongation, that is a protein connect to the correct sequence of the nucleotides into the continuous new strand DNA. And termination, that is a protein release replication complex. Okay, then proofreading new data, this is a DNA polymerase makes about 1 1 in 1000 base pairing errors, and enzymes proofread and correct those mistakes, and the new error rate of the for the DNA that has been proofread in is 1 in 1 billion base pairing errors. Okay, then transcription is the process copying DNA to RNA. Differs from DNA synthesis is that only one strand of DNA and template strand is used to make the mRNA. Does not need a prior to start. Okay, then here is also this process like annotations, then intentions, elongations, terminations. There are the three. Okay, and the types of RNA molecule, that is the mRNA, messenger RNA, transfer RNA, and ribosomal RNA. And translation, the component required for the translation, that is the mRNA, ribosome, tRNA, amino acyl tRNA synthesis. And translations means the process that is copying copying to DNA to DNA to RNA. Okay, and then that is okay, so in today's video we can discuss about the central dogma of the life. So thank you.